Welcome to the Sans Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump in, please take a moment to like this deep dive and subscribe to the channel. It truly helps support the Sans Science Experience. Okay, so today we're unraveling a truly groundbreaking paper. It's by Sauer et al., and it's called Alternate RNA Decoding Results in Stable and Abundant Proteins in Mammals. This research really challenges some basic assumptions, doesn't it, about how proteins get made?
It really does. We're talking about a hidden layer of diversity.
Yeah.
They looked at a ton of data, over 1,000 human samples, healthy tissues, different cancers.
Right. And the mission here is to explore how our cells might be creating, well, completely new protein versions, not just from DNA changes or the usual RNA edits we know about.
Exactly. It's this idea of alternate translation.
Alternate translation. So what does that actually mean beyond the standard picture?
Well, traditionally we think amino acid changes in proteins come from two main places: mutations in the DNA sequence, or maybe some specific RNA editing events. But this paper points to a third way. It suggests the machinery that reads the messenger RNA, the ribosome, sometimes deviates. It swaps out an amino acid.
Even when the genetic code, the mRNA sequence itself, says to put in the standard one.
Precisely. It's like a non-standard reading of the code during the translation process itself.
Okay, and here's the part that really grabbed me. This isn't just some, rare glitch.
Not at all. That's the surprising thing. They found tens of thousands of these events. We're talking 60,803 specific signals.
Wow.
Which boil down to over 8,800 unique substitution sites. And this is across nearly 1,800 different genes.
Almost 1,800 genes. And the kicker is, some of these alternate proteins are actually more common.
Yes, that's the core finding, really. For hundreds of proteins, these alternatively translated versions are more abundant than the standard ones coded directly by the gene. They use this measure, RAS, the ratio of amino acid substituted peptide,
right, RAS,
to its non-substituted version. And for many, that ratio is greater than 1.
So the alternative is actually the dominant form in the cell. That kind of flips things on their head, doesn't it?
It really does. Which leads to the big question, why? Why are these alternate proteins, these SAAPs as they call them?
SAPs, okay. Substituted amino acid proteins.
Right. Why are they so abundant? The study gives us some clues. A big one seems to be protein stability.
So they just last longer.
It looks that way. Many SAPs, especially the really abundant ones with high RAS, were found to have lower degradation rates. They just stick around longer. They aren't cleared out as quickly.
Okay, that makes sense. Stability leads to accumulation, but is it just stability or is something else influencing why these substitutions happen in the first place?
Good question. The paper suggests it's not random noise. There seem to be RNA-related mechanisms involved. Things like how transfer RNAs, the molecules that bring the amino acid, pair up with the mRNA code. It turns out that less frequently used codons might actually lead to more substitutions.
Huh. That's counterintuitive.
A little, yeah.
Okay.
And also certain chemical modifications on the RNA itself, like something called pseudoregulation. These modifications often show up right at these substitution sites.
So it implies some level of maybe regulation, or at least factors that make these sites prone to alternate translation. It's not just random errors.
That's what it strongly suggests, a kind of underlying cellular logic we haven't fully appreciated.
Which brings us to function. If they're stable, abundant, maybe even regulated, what are they doing?
Right, the million-dollar question. Well, they found these SAPs cropping up in really important protein groups, things like transcription factors, which control genes, proteases, signaling proteins, critical jobs.
Okay, so fundamental cellular processes. Any links to disease?
Yes. They noted connections to proteins implicated in neurodegenerative diseases. TDP43, FUS, VCP, names that might ring a bell for some listeners. And they saw patterns. Some SAPs were more abundant in specific tissues or significantly more abundant in certain cancer samples compared to healthy tissue.
Suggesting roles in tissue identity or maybe even driving disease.
Potentially, yes.
Yeah.
And it's not just us. They found this phenomenon conserved in mice, too. So it seems like a widespread biological strategy.
It really broadens the whole picture of the proteome, doesn't it? All the proteins at work in our cells.
Absolutely. Far more diverse than we perhaps assumed based purely on the genome sequence.
So wrapping this up, we've taken a deep dive into how this alternate RNA decoding is kind of silently creating this huge diversity in our proteins. It's just another reminder, really, that biology is always more complex, more ingenious than we think.
Definitely. This research suggests these aren't just mistakes or noise. They're stable, often abundant proteins, and they likely have real functional roles we need to understand.
Makes you wonder, right? What other layers of regulation or maybe disease mechanisms are hidden in plain sight within this alternative proteome? Something to think about. That's all for this deep dive. Please remember to like this video and subscribe to the channel for more insights from the cutting edge of science. We really appreciate your support for the Science experience.